Good morning, students. Production of secondary metabolites. We know plants produce thousands of sophisticated chemical molecules. These include the chemicals which are required for plants' basic metabolic process. Basic metabolism is like nutrition. We call them as primary metabolism, essential for the organism to continue in its living condition, and the metabolites are known as primary metabolites. The chemicals or the metabolites in basic metabolic processes such as sugar, lipid, amino acids, nucleic acid, all are just like metabolites in human being. Whereas at certain conditions, the plants produces some additional compounds. These additional compounds are known as secondary metabolites. These additional products, secondary metabolism products, includes alkaloids, resins, tannin, latex, many more. Why we are very much curious about secondary metabolites is they have a very good pharmacological effect as well as some toxic effects in human being. These secondary metabolites are as a result of secondary metabolism which arises due to the infection in plants, or they may be due to defense or to attract some pollinators, or maybe due to some primary immune response or secondary immune response in plants. But these are of having high medicinal value in human being. Sometimes they may be toxic to human being also. The function of secondary metabolites are not clearly known. They have been implicated in defense mechanism of the plants against pest, pathogen as well as feeding by animals. However, many of these secondary products, especially various alkaloids, are of immense use in human being as medicines. Now these secondary metabolites are obtained through plant tissue culture. We culture the cells, and to these cells we induce some stress signals. It could be nutrient deficiency or any type of stress we give to these cells. The cells are in the form of suspension and they are given some stress. These stress will release some phytohormones and they will start producing secondary metabolism. As a result, we can see various secondary metabolites. These metabolites will be released in the medium and which can be isolated. Why we give stress to the plant? Normal condition plant do not produce secondary metabolites. So we have to induce some stress signal in the plant tissue culture medium, and thus that stress signal will release secondary products which can be purified. So this table shows some examples of industrially important plant secondary metabolites and which are produced through plant tissue culture. Artemisinin, used as an anti-malarial drug. Azadirachtin, which is produced from neem trees, they are used as an insecticidal agent. Berberine is having some antibacterial or anti-inflammatory use, produced from Coptis japonica. Capsaicin, the chili, Capsicum annum, they have some secondary metabolites which are having role in rheumatic pain treatment. Codeine from papaver species, they are analgesic. Digoxin, Digitalis lanata, cardiac tonic. Diosgenin from Discorea deltoidea, anti-fertility drug. Scopolamine from Datura, it is used as anti-hypertensive. Quinine from Cinchona officinalis is an anti-malarial drug. Chiconin is from Lithospermium erythrorhizon, is an antimicrobial as well as used in the dyes and lipstick. Taxol from Taxa species is anti-carcinogenic, and vincristine from Catharandus rosea, that is anti-carcinogenic. So in total we can see majority of the secondary metabolites, they have role in human medicine. Some are used in industry. Discriminative use of such valuable medicinal and other plants has brought them to a near extinction. So another possible solution for this is you can produce these secondary metabolites through plant tissue culture instead of obtaining them directly from the plants. Some secondary metabolites are produced in the roots. So to take out these metabolites, we have to uproot the plant, and that may lead to extinction of the plant. So we can generate these secondary metabolites in large fermenters or bioreactors. Next application is somaclonal variation. See, it is observed that long-term callus culture and cell suspension cultures and plants regenerated from such cultures are often associated with some genetic variations, or these variations in the plants that have been produced by plant tissue culture is known as somaclonal variations. This somaclonal variation is introduced to us by Larkin and Scowcroft in the year 1981. Here picture shows the schematic representation of somaclonal variation. Take the explant from the plants and we grow them to form callus. Certain conditions we form suspension culture. We induce plant regeneration. Some other time we will go for protoplast formation, and all these things. At the end we get plantlets from that. So during all these processes there are some chances of genetic variation. When the cell divide, the cell switches from a differentiated cells to undifferentiated. Then again at the time of formation of the plant's parts or organs, tissues, they again get differentiated. So this de-differentiation and redifferentiation, all these things will lead to some DNA damage and repair. That DNA damage and repair will lead to some variation. Those variation may be harmful as well as useful, are known as somaclonal variations. Somaclonal because we use somatic cells. The tissue from which the variants have been obtained is having gametophytic origin. It means they are from egg or pollen. Then such variations are known as gametoclonal variation. Okay, thank you.